Now we are going to talk about different methods of sterilization. It can be wet heat sterilization, dry heat sterilization, use of certain chemicals, use of alcohol, use of radiations और use of different filters. लेबोरेटरी में काम करते हुए हमारे पास कुछ बहुत ही छोटे पीस इक्विपमेंट होते हैं. लेबोरेटरी में काम करते हुए we have sometimes very small piece of equipment like micropipettes. We work on the benches which are quite big. So we have to make sure that which method is used for which particular equipment. In case of wet heat sterilization, within the lab, we come across with the growth media, glassware, small piece of equipment or laboratory benches. While working in the lab, wet heat sterilization is most common method to sterilize small piece of equipment as well as media, the culture media. में एक प्रेशर कुकर यूज किया होगा. प्रेशर कुकर और wet heat sterilization. The function or the principle of both the methods is same. In this case, what we do, we increase the temperature. In a closed container, there is present water which is boiled to produce the steam. Steam is going to exert some pressure. So increase in temperature plus pressure because of steam is going to make things sterile. In wet heat sterilization, the temperature which is achieved is 121 degrees centigrade and the pressure is 15 psi. And this situation is maintained for 15 to 20 minutes. In wet heat sterilization, when you are going to sterilize the growth media or any liquid, make sure that it should not be tightly sealed in the containers. It should be loose enough so that the boiling water or boiling solution should have some space in the container. Besides wet heat sterilization, we have another method of sterilization which is called as dry heat sterilization. We in the lab have different types of ovens which can go up to 200 degrees centigrade. This 200 degrees centigrade temperature is sufficient enough to give you sterilized conditions. This method is mostly used for the metallic equipment pieces like scissors, some dissection operators, or we can also use this method for glassware sterilization. But keep in mind, if you have to sterilize the glassware, remove the plastic covers or plastic caps of the bottles. Plastic may not be able to withstand such a high temperature for 1 to 2 hours. Then we have certain chemicals like strong acids, strong alkalis, phenols, ethylene oxides, which can be used to make the benches or equipment sterilized. However, this method is not preferred for the sake of safety, of the researcher. Obviously, you will not be interested to work with a strong acid and cleaning a bench or cleaning a equipment or piece of equipment. Within the biotechnological lab, we may have to use glass rods or inoculating loops to take sample or to take inoculum from the bacterial culture or microbial culture. For that purpose, these glass rods or inoculating loops should be sterilized. And that is achieved by first dipping these two things in 70% ethanol and then heating them on the flame until red hot. When you work in the lab, you work on different benches. These are quite big in size. So you may need to clean these places using 70% ethanol. So always start your work and end your work with this procedure. Clean the space with 70% alcohol before you start and clean it after completing your work. It will ensure the sterilization of the working area. Besides these chemicals, we have some radiations which can be used for sterilization, like UV light, like gamma rays. These radiations are most of the time used or installed in the big benches to kill the microorganisms and to ensure the sterilized conditions. In case if we have to sterilize very minute volumes of liquid, for that purpose we can use the filters. These filters have 1 nanometer pore size, and this size is sufficient enough to restrict all the bacteria or microorganisms to pass along with the solution. Depending upon the volume, we can decide about the size of the filter which can be used. Make sure to use appropriate method to sterilize the appropriate equipment. You cannot autoclave a working bench. So, while working in the lab, everything should be sterilized. Autoclave everything by yourself. Don't trust others. Make sure the bench is sterilized. Keep in mind, outside of the bottle, outside of the bags are not sterilized. You yourself is a big source of contamination, so be prepared to avoid contamination. Relax. These methods are time taking. Shortcut means contamination. So avoid shortcuts while working in the lab.